Dear students, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be going over some of the disorders that are associated with purine metabolism. So in the previous lectures that we have seen, de novo purine biosynthesis and de novo pyrimidine biosynthesis, and then salvage pathways for purines, and also we have seen ribonucleotide reductase enzyme and various drugs and regulatory mechanisms we have seen. Now, disorders that are associated with purine metabolism. So, I am going to go over one of the most important disease that is covered in the exams is the Lesch-Nyhan syndrome. And then I will be taking you into 𝗠𝐫𝐫𝐞 syndrome and then into adenosine deaminase enzyme deficiency. And also in the initial part of this particular lecture, I will be explaining you a little bit about hyperuricemia that leads to gout. Let's get into the details. Alright, so increase in the degradation of purines, it will give rise to excess uric acid in the blood and this will lead to hyperuricemia. Hyperuricemia is excess uric acid in the blood is referred as hyperuricemia. And the causes for hyperuricemia can be grouped into overproducers of uric acid and undersecretors of uric acid. Undersecretors of uric acid, it is more of renal failure issues, whereas the overproducers of uric acid, it may fall into metabolic disorders. Like overproducers of uric acid may be because of genetic defect in PRPP synthetase enzyme. That means whenever PRPP synthetase enzyme activity is increased, it will lead to excess levels of PRPP, which will promote purine, de novo purine biosynthesis, and also de novo pyrimidine biosynthesis. But de novo pyrimidine biosynthesis, like pyrimidine degradation products, are water soluble, whereas purine degradation product is uric acid. So that will lead to hyperuricemia. And also if there is a defect or deficiency in HGPRT enzyme, that is hypoxanthine guanine phosphoribosyl transferase enzyme, which is a purine salvage enzyme. Defect or deficiency of this enzyme will lead to Lesch-Nyhan syndrome. So absence of HGPRT will lead to Lesch-Nyhan syndrome, or deficiency of HGPRTase enzyme will lead to 𝗠𝐫𝐫𝐞 syndrome. Okay, so deficiency or absence of either of the absence of this enzyme, it will give rise to excess uric acid formation by de novo pathway. So that will lead to hyperuricemia. Glucose 6-phosphatase enzyme deficiency, that is Von-Gerke disease. In Von-Gerke disease, where glucose 6-phosphatase enzyme is deficient, leading to glucose 6-phosphate, which is diverted into ribose 5-phosphate, and that ribose 5-phosphate going into PRPP and elevated PRPP, leading to elevation in purines, and elevated purines means elevated uric acid, giving rise to hyperuricemia. So, and also hyperuricemia can be secondary to certain diseases like cancer, psoriasis, or it may be chemo radiation, where increased number of cells are died, and that leads to turnover of the nucleic acid, leading to hyperuricemia. So overall, a hyperuricemia will lead to monosodium urate crystallization, that is MSU crystals, monosodium urate crystals, which will show birefringence under polarized light, and they are needle shaped crystals which can lead to inflammatory in the synovial joint cavity. So it leads to gouty arthritis. So treatment for gout includes ibuprofen, diclofenac, colchicine, and also allopurinol. Allopurinol, it inhibits xanthine oxidase enzyme, thereby it decreases uric acid formation, and also it is going to have an indirect effect on de novo purine biosynthesis pathway. Now let's get into Lesch-Nyhan syndrome. Lesch-Nyhan syndrome is an X-linked recessive condition. That means males are the ones who are affected, females act as a carrier of this particular disease. So it is because of absence of HGPRTase enzyme, that is hypoxanthine guanine phosphoribosyl transferase enzyme, which is a purine salvage enzyme. So the absence of this enzyme leads to Lesch-Nyhan syndrome. So it is a disease of purine salvage, where adenine is converted to adenosine monophosphate, and guanine or inosine converted into inosine monophosphate or guanosine monophosphate. So these are all purine salvage pathways where HGPRT is involved in the pathway. So absence of HGPRT lead to defect in the purine salvage pathway, and that may lead to increase the de novo purine biosynthesis, because when the salvage pathways are absent, so purine has to be synthesized by de novo pathway, and that will take a lot of time and energy for, especially for neurons, and it may lead to so many neurological issues. So, and also when HGPRT is not working, so there will be rise in PRPP, phosphorybsyl pyrophosphate, because conversion of hypoxanthine is converted into inosine monophosphate, where PRPP getting into the reaction and making inosine monophosphate. Similarly, guanine converted into guanosine monophosphate, where PRPP is getting into the reaction. So these two reactions catalyzed by HGPRT enzyme. So absence of HGPRT will lead to elevation of PRPP, and this PRPP elevation, which will have a positive effect on PRPP glutamyl amidotransferase, which is the most regulated enzyme and rate determining step in de novo purine biosynthesis. So that will lead to overproduction of purine nucleotides in de novo pathway, and that will affect the neuronal metabolism, because the de novo pathway purine biosynthesis, it needs more reactions and also more ATP. So more time and ATP are spent here by the neurons to make purines by de novo pathway. So that will alter neuronal metabolism, giving rise to neurological issues. So what are the neurological issues we are going to see here? Changes in the neuronal metabolism will lead to bizarre syndrome of self-mutilation. Patient will show self-mutilation, basically harming in their own body parts, like biting the fingers, biting whatever, the lips, biting their arms. So they usually will be tied to the chair, so that they don't cause self-mutilation. Self-mutilation is the bizarre sign that is seen in Lesch syndrome patients. Patients will show decreased IQ. Neurological signs like chorea, athetosis and spasticity can be seen in Lesch syndrome patients. Along with this, patients with Lesch syndrome will show hyperuricemia, uric acid crystallization and uric acid crystals in the urine, which is referred as orange sand in the urine, and uric acid crystal can be found in the kidneys, leading to nephrolithiasis, obstructive uropathy, where if the uric acid crystals are found in the urinary tract, somewhere in the urinary tract, and eventually it can lead to renal failure and gouty arthritis. All these are the signs that we see in Lesch syndrome. Remember that. Coming to Kelly-Sieg-Miller syndrome. Kelly-Sieg-Miller syndrome is because of partial deficiency of hypoxanthine guanine phosphorybsyl transferase. Hyperuricemia is seen here, but generally neurological issues, no central nervous system signs are seen, like no self-mutilation, chorea, athetosis, spasticity, that kind of signs and symptoms we don't see here. So treatment with allopurinol in both the condition will elevate the hyperuricemia signs and symptoms, but generally neurological issues are not reversed. Coming to adenosine deaminase enzyme deficiency, ADA deficiency. Note that adenosine deaminase is the enzyme that converts adenosine into inosine in the purine salvage pathways, and inosine is converted into hypoxanthine, and hypoxanthine can come back into inosine monophosphate formation in the purine salvage pathway, or it can just simply go into uric acid formation by degradation process. So deficiency of adenosine deaminase enzyme, so because of defect in ADA gene, it will give rise to a disorder called severe combined immunodeficiency. 50% of cases of severe combined immunodeficiency is because of ADA enzyme mutation, that is ADA gene mutation leading to adenosine deaminase enzyme deficiency. So for that matter, severe combined immunodeficiency disease, the causes for them can be divided into two groups. One is autosomal recessive cause, that is ADA gene mutation is one of the cause, and other causes which fall under autosomal recessive cause for SCID is interleukin 7 receptor alpha gene mutation and RAG1 and RAG2 gene mutation. These are all autosomal recessive mutation that may lead to severe combined immunodeficiency, that is SCID. Okay, ADA gene mutation, interleukin 7 receptor alpha chain gene mutation, and then RAG1 and RAG2 gene mutation. Coming to X-linked recessive inheritance pattern, it is because of a defect in the gene that is coding for gamma chain of interleukin 2, 4, 7, 9 and 15. So these are the causes for SCID, that is severe combined immunodeficiency disease. So let's concentrate ourselves with adenosine deaminase enzyme deficiency. Though almost all cells are affected here because they are expressing, almost all cells expresses adenosine deaminase enzyme, but lymphocytes are the ones which have got highest activity of adenosine deaminase enzyme. So that means deficiency of ADA, adenosine deaminase enzyme deficiency, will worstly affect lymphocyte unlike all other cells, because ADA deficiency will lead to accumulation of adenosine and deoxyadenosine, especially in the lymphocytes, because other cells, they will secrete adenosine and deoxyadenosine into the blood, whereas the adenosine and deoxyadenosine secretion is done in lymphocytes, both T lymphocytes and B lymphocytes. In fact, they will take up adenosine and deoxyadenosine from the circulation, and a buildup of adenosine and deoxyadenosine in these cells, what happens is, so this adenosine will be converted into adenosine monophosphate into adenosine diphosphate into adenosine triphosphate on one side, and this adenosine diphosphate, some of it undergoing ribonucleotide reductase reaction, becoming deoxyadenosine diphosphate, which is converted into deoxyadenosine triphosphate. So that means there will be increase in the ATP and also increase in DATP in this particular disease. So especially increase in DATP, it will have an inhibitory effect on ribonucleotide reductase, because DATP is an inhibitor of ribonucleotide reductase. So whenever there is elevation of both ATP and DATP, so the inhibitor DATP, which will predominate, and it is going to inhibit ribonucleotide reductase enzyme, thereby it is going to decrease DGTP formation, DTTP formation and DCTP formation. So all deoxynucleotide triphosphates, other deoxynucleotide triphosphates will be decreased, while deoxyadenosine triphosphate will be elevated. So that leads to overall decrease in the DNA synthesis and also decrease in the RNA synthesis. So this will affect both T lymphocytes and also B lymphocytes. So both T lymphocytes and B lymphocytes will be decreased, their proliferation will be decreased. So that's why patients will suffer from combined immunodeficiency disease, that is both the humoral immunity and cell mediated immunity will be deficient here, and that's why these patients will show opportunistic infections like fungal infection, bacterial infection, protozoal infection, all kinds of infections can be seen, and usually they will succumb to death. So this is the pathophysiology of adenosine deaminase enzyme deficiency leading to severe combined immunodeficiency disease. So these are some of the disorders associated with purine metabolism. Thank you.